For panel design, I don't want to spend too long on this topic because I think that a lot of the concepts are fairly similar to conventional flow cytometry, but I will go over a quick background on it, and I also want to go over the tools that are specific to the Aurora and how to use those. Before you design a panel, it's best if you gather as much information as possible. So try to understand the biology of what you're doing, what has been published previously using the markers that you're utilizing, and then ideally if you can classify your antigens into primary, secondary, and tertiary. If you hadn't heard this terminology, I'm going to explain this on the next slide. And then also understand the antigen co-expression, which is very important for designing a panel. So the antigen classification, primary is your on or off markers. So examples of that would be CD3, CD4, CD45. You can see here in this plot the CD3 on the x-axis. It's got a very clear positive and clear negative population that are separated, so there isn't really any in-between staining. Whereas with a secondary marker, that's more of a continuum. So some examples are CD45RA and HLA-DR. So you can see on the y-axis, I have the HLA-DR. You can see I would call this more of a smear. So this is what would be called a secondary classification of the marker. And then the tertiary would be your low or unknown expression. So a few examples might be FOXP3 or ICOS. And if you are unsure about the expression, then I would recommend doing some searches, try to figure out what's been published, what you might expect these markers to look like if you can. Hopefully you don't have a panel that is entirely tertiary markers. Hopefully you do have some primary and secondary markers and only a few tertiary markers. Then for antigen co-expression, there's a few questions that I recommend you think about. So what markers are you using to define or identify your populations versus what markers characterize these populations? Something that you might be looking at, for example, maybe activation status. And then it's also nice to think about what markers are on a lot of cell types versus a small number of cell types. So CD45, if you're looking at immune cells, is going to be on all of your cells, whereas in whole blood, CD25, for example, is going to be on a small percentage of these cells. So knowing this information can really help place the markers in the right fluorophores. And then if you talk to SciTech about designing a panel, or you can also talk to me about designing a panel, or a lot of the reagent companies like BioLegend or Thermo Fisher, they can help you with panel design as well. They may ask you for some additional information about the populations that you're interested in, and there's a few different ways that you can summarize this. So you could draw out some plots and show where you may expect to find populations. I know that a lot of people are interested in the unsupervised analysis tools like t-SNE, but it is still really helpful to think about these biaxial plots when you're doing panel design. Another way to do it is you could do like a hierarchy like this that shows where all of the expression is. You may find some papers that have published data. This can be extremely helpful for panel design to see what data would actually look like. And a lot of times papers will publish full gating strategies. So if you have information like this, this could be really useful for panel design because it really tells us, which markers are on-off, which markers are continuous, which populations are rare. And all of this will factor into how you design a panel. A few other considerations that you need to make. So first you have to decide how many fluorochromes you're going to use in your panel. And that depends on how many antigens you want to detect and therefore which fluorophores are the best for that number of antigens. So I will tell you, if you have a smaller panel, so I don't know, maybe 10 markers, then it would be best to design that so you are not utilizing fluorophores that excessively spill into each other. or are very, very similar to each other. So for example, if you have a smaller panel, like an 8-color panel, do not put BV421 and eFluor450 in that 8-color panel, because you have a lot of other options with fluorophores that aren't quite so similar. As I mentioned, just because we can separate these fluorophores doesn't mean that it's easy to separate the fluorophores. So save the very similar fluorophores for when you are designing the larger panels and maxing out the capabilities of the instrument. And if you don't know which fluorophores are similar, they're basically the fluorophores that you would not be able to separate on a conventional flow cytometer. And then finally, you also need to think about which antibodies are commercially available. Sometimes antibodies only come in a few fluorophores, and there's also some fluorophores that are not very commonly used. For example, Alexafluor 532, there's only a few antibodies that are available in that fluorophore. And then also think about clones, because you can get different staining pattern with different clones. So pay attention to clones, pay attention to which antibodies only come in a few fluorophores. So to assign the fluorophores, if you have a panel that's similar to Cytek, so this is the one that they have tested extensively and works for immunophenotyping PBMCs, you can start with this one, take out the antibodies that you're not interested in, and then replace those with your antibodies. That's one possible way of designing panel. Or if you want a totally different panel, then I would definitely recommend using the tools that are specific to the Aurora. So that includes the spread matrix, which I'll talk about later. And then there's also on Cytek's website or on our website in the references section, you can design the Fluorochrome selector, and it will include the brightness indexes for all of the fluorophores, because you do want to still pair bright fluorophores with low express markers and high express markers with dim fluorophores. So take a look at the brightness index for the Aurora specifically, and then also you definitely want to keep in mind the spread matrix. So to better understand the spread matrix, I'm going to talk a little bit about what spreading is and what spillover is, because they're related but different. So spillover you've probably heard of if you have done a lot of conventional flow cytometry. So on this top graph, we've got our emission spectra for AF700. Then we've got the AF700 detector in blue. And then you can see that there's a little bit of APC in this detector as well. So we say that APC spills into the AF700 detector. Pretty straightforward. With with spectral cytometry, it gets a little more complicated because we do have so many more detectors, and there are obviously quite a few detectors that have multiple fluorophores in them. This is only 2 colors, but you can imagine as you add colors, then a lot of the detectors are going to have quite a few fluorophores in each detector. So we would visualize the spillover like this. So this is a different example where BV711 is spilling into BV421, at least that's what we would say in conventional flow cytometry. And we would also say that this plot is undercompensated. So with compensation, we would move this positive population back over here so that the center of both of the populations would be aligned. And so the compensation would fix the spillover. In spectral cytometry, undercompensated is obviously not the right term because we're doing spectral unmixing, not compensation. Under unmixing is a bit of an odd term, so I believe that Cytek has started saying this is undercorrected, and the other direction would be overcorrected. And then as far as BV711 spilling into BV421, it's also kind of a weird way of phrasing things because we don't really have an assigned detector, but there isn't really any other terminology. So now spreading or spillover spreading, you will see you will see when the compensation has been applied. So here's another example where we've got an undercompensated sample, or uncompensated sample, which also happens to be, I guess it's undercompensated. So we apply compensation to this, and ideally we would hope that our sample now looks like this, but in reality we see something like this. We see spreading in this positive population. And you may have encountered this in conventional flow cytometry because it definitely happens both there and in spectral cytometry. So you may be wondering, what is this spreading? Well, it occurs from errors in measurement and data binning and also photon counting statistics. Basically, it's a bunch of complicated physics, and I'm not a physicist. So my simplification of that is that there is measurement error when other fluorophores emit light in the same detector, and the photons are a little bit difficult to count, so there's some error in counting them. And so we get this variability in the population, and it ends up spreading. Now, one thing to note about compensation or spectral unmixing is that it does not introduce or increase the spreading error, it only reveals it. So the error is always going to be there. It's just that once you have applied compensation or once you have done the spectral unmixing, it just becomes much more obvious that there is a spreading error. So this is one reason for needing staining controls that helps you decide where to place the gates, especially when they're spreading error. But if you do want to do some more reading, there are some links down here. And the main reason why we need to worry about spillover spreading has to do with resolution of your population. So keep in mind that the more fluorophores you have in a panel, the more you see the spreading error. Again, both in conventional and spectral flow cytometry. And what happens is we have an example here where we have a single stain for BV785. We have a nice positive population and a negative population. But when I added in 13 more colors to the panel, I've now lost the resolution of my population. I can't gate that population that was nice and clear in the single stain. So this spreading error is causing me to not be able to gate on my population of interest. Now we do know a few things about spillover spreading because, as I said, it happens in conventional flow cytometry. So more spreading error occurs with brighter intensity. So you can see here that as the fluorophore, this APC-Cy7 fluorophore gets brighter, it spreads more into the secondary channel, so the Q.800 channel. So if APC-Cy7 was dim, then we can draw the negative for Q.800 at this level. But if APC-Cy7 is brighter, then we need to draw our negative at this point. More spreading error also occurs with more fluorophores in the detector. So again, I think this is a little bit easier to wrap your head around if you think about conventional flow cytometry. So we have our filter for BV605, and you can see that I have a few other brilliant violet dyes on this plot as well, and they're all spilling into this BV605 filter. So the more fluorophores that you have spilling into this filter, the more spreading error is going to occur. And then also, the more error you're going to get with a fluorophore that spills more into a detector. So if you look at this green line, that's spilling into the detector a decent amount, but compared to the brown one, that one is not spilling quite as much. So the green one is going to cause more of a spreading error than this brown one is. So here is an example. Again, this was run on our X20 conventional flow cytometer. If we look at the spectra for PerCP Psi 5.5 and BUV 737, you can see there's some overlap. They're pretty close to each other. We can look at the compensation values. This isn't exactly the most accurate way of measuring how these fluorophores spill into each other, but it kind of explains it. So they're fairly high compensation values, and then if we look at the two colors against each other plotted, we can see that there is some spreading of the population. So they're a bit on the wide side. And that becomes very apparent when you compare this instead to BUV 805, where there's very low compensation, there's not a lot of spilling into each other. And so compared to when we looked at BUV 737, which has lots of spreading, the BUV 805 does not have nearly as much spreading. Now, how do we fix this or minimize this? Remember that the spreading error is always going to be there. So what we need to do is design our panel around the spreading error. So the easiest way to deal with lots of spreading is to think about co-expression. Here's an example, again, conventional flow cytometry. You can see the blue population is spreading quite a bit. And let's say that our population of interest is this red one. If we've got it on this fluorophore with BB711, this spreading error is okay because we're still able to resolve our double positive population. But if we've got it on this other fluorophore, BB605, we're still getting spreading error. It's actually a little bit less than this one, but now we can't resolve our double positive population. So this setup would be an issue, whereas this pair of fluorophores would work fine for the markers that they're on. So the best way to deal with spreading is to plan out so you don't have to worry about the double positive population. So that's what I've done in this graph where I looked at CD66B on the granulocytes and CD8 on the lymphocytes. I don't expect any double positive cells because they're completely different cell types. So I never have to worry about the double positives, and I don't really have to think about how these two markers spread into each other. So if I have two fluorophores that do spread a lot into each other, then ideally I put them on different cell types and I don't have to worry about it. If I have to put them on the same cell type, then I need to actually do an experiment and see if I am able to resolve my population. Another way is to use dim markers because remember, the brighter the marker is, the more spreading it causes. So if there is a fluorophore that causes a lot of spreading, then I try to put that on something that is a bit dimmer. And then also just a reminder to titrate your reagents because by titrating them, you can get them to their dimmest possible intensity. And so you're going to get better results if you don't have an excess of very bright fluorophore. If you could get by with a less bright fluorophore or a lower concentration. So now coming back to the spread matrix, this was given to us by SciTech. So you can see to read this table, the spread of the fluor in the row impacts the resolution of the fluor in the column. So here's an example, PE Dazzle 594, PE Sci 5. You can see there's quite a bit of red in these two rows. So this tells us that these two fluorophores cause quite a bit of spreading in all of these different fluorophores. So I would not recommend putting something like CD45 on either of these markers because it's really bright, it's on all of the leukocytes, and it's just going to cause lots of problems. But a nice thing to put on here would be a dump channel or a lineage channel because then I don't care about any of the spreading because none of my cells of interest are going to be positive for this marker. A thing to keep in mind about this matrix is that it was created with all CD4 reference controls for all of the fluorophores. So this is sort of a worst case scenario. And if you plan out your panel carefully, then you won't actually get this level of spreading. And then finally, it also assumes that all of the markers are within the tube. So if you're not planning on doing a 24 color panel, let's say you're just doing a 15 color panel, then you're obviously going to get slightly different results because you don't have all these 24 colors. So there's a few different ways to assign the fluorochromes once you have decided on what markers you're using and taken a look at the spread matrix. You can start with your tertiary antigens because those may have the most limited options. Then you can place your secondary antigens, but you want to be wary of co-expression. And then finally, you can place your primary antigens, because usually those primary antigens come in a lot of fluorophores, so they should be relatively easy to place. Or if you want to, you can start with the easy ones, the primary antigens, and then work your way backwards. For live dead dyes, I do always recommend including a live dead dye because you will get better results. SciTech tends to recommend the Zombie Near IR dye from BioLegend. I have found that if you do not titrate this down, you may find that you're getting some issues with AF700 and APC size 7, which are similar to this Zombie Near IR signature. So definitely make sure that you titrate the Zombie Near IR dye down. If it's too bright, you could have issues. And as I mentioned, it's best to manufacture this control, so make sure you kill cells and you definitely have a really bright, very dead population of cells. You can use the other dyes like DAPI and PI. Just know that you really need to titrate those because the concentration that you're using on any of the other instruments, like our Fortessas, it may still be too bright for the Aurora. So definitely titrate these down, but you can use them for live dead. If you want to use these for cell cycle, that becomes a little bit more challenging, and you should definitely talk to me about that.